Can you see that? Can you hear me okay?
Yes. All right. Yes.
Okay. Yeah, so hi, my name is Christina. I've actually kind of moved around a bit. I was at the University of Akureyri last year. I'm now an assistant professor at St. Mary's College of Maryland. And then I'm also a research scientist at the University of Maryland Center for Environmental Science. So I've bounced around a bit, but I'm back in the US now. And all of my work is looking at kind of the effects of climate change on benthic populations in the Arctic, and I'm really interested in temperatures. So I'm coming at sustainability from a little bit of a different direction, thinking about the sustainability of these populations and are they able to survive through these changes, and then kind of at a larger picture what that means for communities that rely on them as a food source or maybe rely on the animals that then feed on these animals. So that's kind of where I'm coming from and what I'm going to be talking about today. So all of my work is in under this umbrella program called the Distributed Biological Observatory, and the goal is to kind of link the physics of the system with the biology. It was first proposed back in 2009, and we had a pilot year in 2010. Since then, we have multiple national and international cruises that go out seasonally and yearly. And we've created this network of what we call transect lines, and treating it as a change detection array. So a group of stations that are then bound by these boxes. So the boxes being for people doing things like seabird observing or mammal observing can work within kind of this box rather than at the stationary points where we might be taking environmental data about the physics and the chemistry, and then data about the biological systems and communities that live there. And so it follows the sea ice edge. So these move latitudinally, they move north up the coast of Alaska into the Canadian Arctic and the Beaufort Sea. And the idea is that all of these places are, excuse me, known spots of high rates of productivity, but also areas where we're seeing really high rates of change. So we call them kind of these regional biological hotspots, and they work in this latitudinal gradient. So people collect all kinds of data here, but my interest is in the benthic community, more specifically the bivalves or the clams. So this is just a taste of what one of our samples would look like if we pull it up. This is a very clam heavy area. I'm very biased towards it, so I picked it on purpose, but these are some of the species that I work with on the Pacific side. My time in Iceland, I spent working on Arctica Icelandica from out in the fjord, but these are the guys that I'm usually looking at. So a variety of different species here that I've done some research on, tracking how they respond to these temperature changes and what that might mean. So the overall goal of what I do and kind of these projects is to understand the ability of dominant bivalve species in the Pacific to adapt to their changing environment. So the Arctic is one of the fastest, if not the fastest, changing place on the planet. And so specifically, I'm interested in rising temperatures. And to do this, I look at things like respiration rates, or how fast they're breathing, in whole sediment cores, so cores that we take out on the ship and are able to kind of mimic an undisturbed environment. I do experiments with individual animals, so looking at differences between species, but also differences within species. So how might one population potentially be a little bit more resilient within themselves, and then looking at important ecological characteristics. So we actually know, at least on the Pacific side, very little about how these animals would exist in a non-changing world. So I've done studies looking at what we call the critical thermal maximum, which is the temperature at which you would start to experience physiological stress or even death. And so if we're trying to answer how they're going to adapt to a changing environment, if we don't know what their ranges are and where mortality would happen with temperature, it's hard to say how they might respond to increasing temperature. So taking that map of stations This map on the right-hand side then breaks those stations down into the dominant organisms that we find there. So the brown dots are the ones that I'm really interested in. Those are the bivalves, which again are going to be primarily clams. We do see mussels occasionally, but the majority of the individuals are clams. So a big hot spot of clams in the northern Bering Sea, and then the southeastern Chukchi Sea just north of the Bering Strait, and then again up in the northern Chukchi Sea. These are areas where you also see a lot of yellow, so those are going to be your polychaete worms. We are actually seeing composition shifts of dominance down here in the northern Bering. We're seeing the emergence of polychaete worms coming in instead of bivalves, which does have implications for the predators there. But broadly, I just wanted to show you that these areas that I'm working in really do have a high benthic component. So I love clams for the sake of the clams, but the reason that they're of such interest in this region is that they are a very common prey item for a variety of predators. So specifically thinking about the spectacled eider, which is a diving sea duck. This is the spectacled eider's range in Alaska. They are a threatened species. So I would just draw your attention to this black box here. This is where the entire known population overwinters. So they all come and feed in this one area, and that corresponds directly with where we're sampling for clams. So they are feeding on the clams. And then the Pacific walrus. So yellow and green are looking at different seasons where we might find females or males feeding. But again, these black boxes are blocking off our regions in northern Bering, the southeastern Chukchi, and the northeastern Chukchi. So walruses are feeding here. They will feed on polychaete worms, but clams are their preferred prey. They can eat upwards of 3,000 to 4,000 of them a day. So they're consuming really large quantities. And then these upper trophic animals, so the birds and the walruses, are very important culturally to the indigenous coastal communities in Alaska. So the interest here a lot of times is it really does trickle just directly up the food web into the human population as well. So we're seeing marked changes in this region. Sea ice is declining. This is annual sea ice persistence. So just the number of days that sea ice was present in a region from 2013 to 2018. Purple and black, so darker colors, are going to be less days where ice was present. And you can see in the northern Bering, we used to have 150 to 210 days of sea ice in 2013. And by 2018, we were looking at anywhere from 0 to 30. That's a huge change. It has a lot of effects both on things like temperature, but also what we're seeing now is an increase in things like storm events because there's more open water, so the wind blowing through. So these coastal communities are also getting slammed with heavier fall storms that they otherwise might not have been used to. In addition to things like ice just being present, it's also leaving and arriving in the area earlier and later. So this dark blue line is the mean from 1981 to 2010. And so generally we see sea ice leave the area completely by June, but return in November. 2012 is the light blue line. This was up until recently kind of the poster child year of climate change in the Arctic. And we had ice not coming back until December, but still leaving in June. So not too badly on the timing, a little bit off the mean, but not too far off. 2018 and 2019 are some of the two hottest years that the Pacific Arctic has been talking about as we've seen changes. So you can see the red line, 2018, the ice came back in December, kind of did a wiggly thing, was here in January, looked like it had completely disappeared by February. That's a huge difference from June. It did spike and come back in March, but then was completely gone in April. 2019, we saw a really similar pattern with the ice not coming back until late December, early January. It did kind of this up and down thing, but again, was gone between April and May, which is much earlier than that June date. So when we talk about sea ice, not only are we talking about the days that it's present and its persistence, but also when it's arriving and leaving an area. So that has a huge effect on the changes for bottom water temperatures. So I'm specifically interested in bottom water because these are benthic critters. So this is our Northern Bering Sea area, and this is it moves northward in latitude by station here. And from 2000 to 2018, you can see consistently the bottom waters here are -1, -2. There's a pocket of cold water here that historically forms every year, but in 2018 and 2019, we saw a huge increase up to 2 degrees. My boss has been working in this area for about 35 years and has never seen this area in the positive temperature. So it was a huge change that we saw in 2018 and 2019. And so we're starting to see these additions of increased temperature in the area, which leads to some of these questions of what can these populations sustain? So one way that warming seawater might have an effect on this system, particularly the benthos, is its availability for food. So as warming seawater temperatures, we could have an increase in zooplankton growth, abundance, and grazing. And why that matters up in the Pacific is we have a generally shallow system. And what happens is our ice edge phytoplankton blooms, so all this plant food material that forms on the ice edge, because it's mismatched, it's showing up generally before zooplankton are going to be grazing upon it, it lands on the bottom. And so we get a lot of really good nutritious food to feed the animals that live in the mud. So right now we have what we call a benthic dominated system that feeds this benthos, is feeding things like gray whales, those walruses, and those diving sea ducks, and then a generally small zooplankton population. Now that's the opposite on the Atlantic side. So on the Atlantic side, we tend to see a much larger zooplankton population and a much larger pelagic fish population. But that's what the concern is for the Pacific, is that we're pushing kind of toward this pelagic dominated future. And so if zooplankton eating increases and starts to match the timing of these blooms, there might be less food that's available to the to deposit to the bottom, limiting that food for the organisms on the seafloor. And now these organisms, their populations are generally driven by the availability of food and temperature. So this is a factor that's really affecting the populations. Oh, sorry about that. So there's also the indication that with changing temperatures, we might see new predators in the region. So more Pacific cod or pollock in the Bering Sea. This is looking at 2010, 17, 18, and 19. And it was a NOAA cod, and it was a NOAA fish survey. This pink blob here in 2010 is that bottom water that I showed previously. So this is really cold water, and you can see the fish both on the cod side and the walleye pollock side show up along that edge. So the temperature is too cold for the adults. It's acting as a thermal barrier that keeps that area safe for benthic creatures and also some larval fish. As we see that pink blob start to disappear, we see fish start to inhabit that area. So the only reason they aren't inhabiting that area is, well, maybe not the only, but the primary reason they're not inhabiting that area is that the temperature is keeping them out. As soon as you warm those waters, those fish start to come in and can take advantage of the prey there. However, that might be pushing out things, again, that are feeding there already, like walruses and birds that are important to the coastal communities. So how do I track these changes and what do I look for? I use experimental work. I do modeling work as well, but I think the experimental stuff's a little bit more exciting. I do this at sea on ships. I go up to the Arctic every year at least once, and I run, this is a kind of a conceptual diagram of our sediment cores. So it comes up, it has undisturbed sediment and hopefully all the critters that are living in it. This is how I would run an individual experiment. So each of these jars has an individual animal inside of it with a probe that's measuring oxygen. And this is what the setup just kind of looks like in the cold room on a ship. And then I've also been looking at these ecological characteristics like critical thermal maximum. So as you steadily increase the rate of temperature, at what point does an animal kind of experience physiological stress or even death? And so I'm looking for, this is that upper critical limit that I'm looking for, but presumably their range and their optimal performance is somewhere lower than that temperature. So this is some data from the core experiments, and broadly what I just want to point out here is that usually the red being the warm years, not always the case. It does depend on what region you are, so there are regional differences, but the red bars or the red boxes are usually, oh, this is a bad example, there's no blue. but they're usually higher than the ambient temperature. So we're increasing the rate of respiration on the bottom in the sediment cores as we increase temperatures. That's not always the case. There is potentially a seasonal signal. So this is September, this is August, and this is October. And you can see October kind of seems to have the opposite problem. So we're still kind of thinking about that data and what that might mean. And are we going to see different signals depending on what time of year we're out there sampling or measuring? If we look at individual animals, these are some of the dominant species I've used. So this white clam here, you can see has a fairly low respiration rate on this side, but if we move up into the southern Chukchi Sea, it increases. So there are just geographical differences even within the same species. But this is just to try and start getting a baseline of what their respiration might be. And then if we start to add temperature, you can see those red box plots are generally a little bit higher or significantly higher than the blue ones. So as we increase temperature, respiration rates in animals in these bivalves are increasing. This is something we would expect to see, but it is nice to kind of see that confirmed in the data. So from some of that work, I've started moving and looking at the resiliency and within species responses. So I've done some stuff trying to ask questions about if you take the exact same animal, so now looking at just this animal's ability to adapt, what happens when you put it in a cold environment and then another cold environment with food and then a warm environment, how do their respiration rates respond? This data is really in its very preliminary form. This was like just the first cut of everything. I don't have any results from this, but trying to think about what is the ability of an individual's capability of adapting, because if you have a lot of individuals that can adapt, then maybe that species might be more resilient as a whole. So we are seeing seasonal responses potentially. Different species are behaving differently. But the same species can behave differently if it's in a different region. So there's a lot of factors going on in how they're responding to this temperature. But understanding how these species respond can help us put it into the larger context as we also see shifts in the community composition. So what species are present over time, and is that directly related to the temperature? So the critical thermal maximum work, they have these responses to temperature, but we know very little about what their temperature range is and what their upper level tolerances are. So an undergraduate student and I, this is her senior thesis, she looked at this, looking both at gaping or indication of death, as well as oxygen consumption, again, respiration rates as a measurement of stress. So she did this both in the field and in the lab. This is her in our cold room back in the lab at CBL running the respiration rates. And so we use, this did not show up this way on my other computer, I'm sorry. We use these fire-sting oxygen probes, looking at measurements of oxygen every 2 minutes, and then looked for gaping of the shell, which would have been an indication of death of the animals. So she had 2 sets of experiments. She had field observations where they were acclimated for 72 hours and then run aboard the ship, and then laboratory observations where the animals were acclimated for 3 weeks in a cold room. And she got the bonus of I've had one of the other species living in this cold room for 7 years. So she was able to use fans that had been acclimated for a 7-year period. And what we found was that the white clam, Lacoma calcarea, its noted temperature range is 0.4 to 10.3 degrees Celsius with an average of 2.7. But I showed you data where they're coming from, the bottom water temperatures in the northern Bering Sea is -2. So this seems to be a little bit of a suspect range anyway. After about 15.69 degrees Celsius, most of the animals were dead. This was after a 72-hour acclimation period, and we started to see respiratory stress, kind of a sharper decline in the respiration rate on average at about 8.36 degrees. So they were dying at 15.69, right? But the ultimate goal wouldn't be to kill all of the animals. That's not the temperature we wanted. We're more interested in where do they start maybe experiencing stress. The animals that were acclimated for 3 weeks, they all survived to about 22 degrees, and we didn't start seeing major respiratory stress on average until about 19.3 degrees Celsius. So acclimation period and the amount of time that they are exposed exposed to their environment and allowed to settle before running these experiments does seem to have an effect on what that might be. And then looking at this final species of Stardy borealis, we didn't actually have any animals die at 22 degrees Celsius, and that was as warm as we could get the refrigerator. We had it unplugged, the door wide open. We couldn't get it any warmer. And so they've been sitting in their kind of artificial environment for the last 7 years. But they also had no visible respiratory stress. But looking at the literature on the species, a lot of their problems are more dictated by oxygen availability and salinity changes. So if the salinity was stable and they had an available amount of oxygen, they might not experience temperature stress, which might make them a hardier and more sustainable animal or species in that composition. So we might see these emerge over some of the other ones potentially. So the conclusions from that were that there were fairly high temperatures that they were able to survive. But again, it is really species dependent, which is important for understanding stressor effects and basic ecological characteristics, because our benthic community is changing. So this is looking at composition only up until about 2010, but you can see some species seem to be declining, others might be increasing. So knowing how they're going to respond is important. So just broadly, like the conclusions and future work from this whole kind of my whole goal. Temperatures is one of the largest drivers in this system, and it's changing. Benthic populations are responding. We're seeing changes in community composition, but it's also bringing new stressors into the region. I didn't really talk about this much, but we're seeing an emergence of harmful algal blooms that are getting into the shellfish. They're finding it in the tissues of the bivalves, so they are seeing it then potentially enter into the predators like the walruses and the birds, which are then being consumed by local communities, and so potentially entering there. Alaska for the last several years has also had paralytic shellfish poisoning warnings about the bivalves off the coast. So themselves as a first point of food source has also had major warnings recently. So future where we're trying to take this is I will still keep doing core and individual respiration work. We have plans for it this summer. But thinking about resiliency and how sustainable this community is what species can survive. Are the ones we're seeing in the community structure the ones that are able to survive? What is the within species diversity? So could the clams of the same species in this region handle it better than the clams down in this region? And if so, why? Taking the critical thermal maximum studies and looking at longer experiments and more species. We've talked about looking at what genes might be expressed, looking at geographic differences. So clams the white clams we were using were collected here, but do the clams down here have a different response? And then how long can those species actually survive at temperatures that are just below where they start experiencing respiratory stress? Our ultimate goal is to know how these populations will survive. So knowing how long, what duration they can handle at these stressful temperatures will start to get at that question a little bit better. So these are all the people, there's been a whole host of people involved in this. And I miss Iceland, so I made the final slide a picture of Akureyri on my walk home from last year. So I'm happy to take any questions.
Okay, thanks, Christina. So questions here on online.